I'm going to be speaking about, deciphering rare diseases and empowering clinical research. This will follow on by a more specialist talk by Dr. Udin later on. So what do I mean by rare diseases? Rare diseases are not rare, unlike what the definition, unlike what it says. I mean, rare diseases affect more than 300 to 400 million people worldwide. So that's roughly 5% of the global population. Uh, More granularly, it translates to about 1 in 2,000 people actually having some sort of a rare disease. And advances in sequencing technology over the years means that we now know of more than 8,000 rare diseases. Most of them that have a genetic origin, about 80% of them have some sort of a genetic origin. Most rare diseases are complex. They cause severe multisystem disorders and manifest themselves with a range of phenotypes. More critically, about 70% of all the rare diseases start in childhood, and this has a major impact on mortality of the children affected with rare diseases. Additionally, in addition to the genomic basis for it, about 23% of the patients that have developmental delays also seem to have an epigenetic association with the rare disease. That is something to do with imprinting, methylation, etc. Now, talking elaborating a little more on rare diseases, about half of the number of rare diseases known affect the nervous system. 90% of childhood rare diseases have neuropresentation, and unfortunately, they are the major cause of mortality in childhood. About 35% of children die in the first year of their life if they have one of these rare diseases. About 900 of these disorders are caused by a single gene and are known to be treatable. However, I did mention there are over 8,000 diseases, which means that, upwards of 7,000 rare diseases do not have a single gene cause or are not treatable. This often means that patients undergo a lot of unnecessary tests and procedures and delays in receiving appropriate care. Most of the time, it takes up to 5 years or more for a rare disease patient to get a diagnosis. In some cases, it has even taken decades. The sad truth remains that many families will never get a diagnosis. I just spoke in the previous slide about the long process. So traditional labs still follow methods that are sort of been practiced for decades before turning to whole genome sequencing. And therefore, it lengthens the time from testing, initial consult, to an answer. So it all starts with a clinical assessment, and then based on what the clinician thinks at that point, patients can go through a series of tests over a period of time. It includes things like Southern blot, long-range PCR, flanking PCRs to detect repeat disorders, then a more targeted gene panel. And if nothing is found, then they start doing things like, FISH or karyotyping to look at suspected copy number variation. Finally, ending up with, microarrays and ultimately a clinical exome. This whole process costs money, it takes time, and causes undue distress to the families that have somebody suffering with a rare disease. When none of this works, after a few years, maybe 5 or 6 years, they turn to short-read whole genome sequencing. And, even after that, about 60% of the cases remain unresolved. In the best case scenario, only about 40% of the cases actually get a diagnosis. Now, this is a problem, given the number of rare diseases and the number of people affected worldwide with rare diseases. What if there was another method to actually shorten this diagnostic odyssey, as we call it? One of the things, this paper by Kim Boycott has been looking at whether long-read sequencing can actually result in additional diagnostic yield. So they did a meta-analysis across various studies, and they demonstrate in this paper that using long-read sequencing as the first port of call for any genetic disorder results in an increase of diagnostic yield by 7 to 17% over the 40% that you've got already. So this takes us over the 50% mark in most cases. Long-read sequencing provides an improved ability to detect structural variation compared to short-read sequencing, better detection of tandem repeat expansions, improved mapping in regions of high sequence similarity, pseudogenes, which I will touch upon in a future slide. Uh, it also allows, in most cases, to assess the methylation profile at the same time as sequencing, thereby being able to shed light on epigenetic markers that cause disease. So that's long read. I mean, long reads are useful, increased diagnostic yield by 7 to 17%. Oxford Nanopore, why, Oxford Nanopore is a long read sequencing platform. It's a single test, which then obviates the need of doing the diagnostic odyssey starting from southern plots onwards to exome sequencing to short read WGS. And a paper recently by Abu Tayoun from Dubai, he quotes long read sequencing technologies could serve as a unified platform for clinical genetic testing, particularly in rare disease settings where nearly half of the patients remained undiagnosed. He demonstrated in a proof of concept study that he was able to get 13% additional genetic disease outcomes and diagnosis compared to short read sequencing, which falls nicely in between what Kim Boycott said previously between 7 and 17% diagnostic yield. Abu Tayoun, Dr. Abu Ahmed Tayoun at Al Jalila Hospital has also been looking at spinal muscular atrophy, a disease that is particularly prevalent in the Middle East due to a highly consanguineous population. He has developed a novel SMA, spinal muscular atrophy test, which can actually take you from sequencing to answer in less than 24 hours. What he's basically done is use the combination of PCR amplification, targeting a specific region of these highly similar genes, SMN1 and SMN2, which are responsible for SMA, and being able to reduce the time to diagnosis by, less than 48 hours, in some cases, even 12 hours, 24 hours. He's been able to multiplex these samples in large quantities on a min-ion flow cell, thereby reducing cost and time taken for diagnosis. If you don't know much about SMN1, SMN2, what you should realize is that they're highly similar genes and they differ only by a single nucleotide in the coding regions. So they are highly similar. SMA, spinal muscular atrophy, is caused by the deletion of the SMN1 gene. And the SMN2 gene copy is only about 10 to 20% as effective as SMN1 genes. So the copy number state of SMN2 actually determines the severity of the disease. So if you have more copies of SMN2, no copies of SMN1, your symptoms are likely to be less aggressive than if you had fewer copies of SMN2 and no copies of SMN1. Another example showing the utility of long-read sequencing is the Beacon project. This is work that was recently presented by Wendy Chung at ACMG 2025. What she showed was an increase in diagnostic yield of about 24% across the cohort. So she studied 350 plus families that were all unresolved by short-read technologies. They come from a range of disease categories, starting from, congenital myopathies and dystrophies, atresia, neurodevelopmental disorders, and so on. And, in the graph on the right-hand side, you can see the percent of increase in diagnostic yield depending on the disease category. Overall, she showed a 24% increase in diagnostic yield using Oxford Nanopore sequencing. So more than the 7 to 17% that Kim Boycott mentioned earlier. So what else can Oxford Nanopore do? So we now know that methylation plays a critical role in rare diseases. What is methylation? This is the selective methylation on certain nucleotides, primarily cytosine. So you get 5-hydroxymethylcytosine or 5-methylcytosine, and these have an effect on the regulation of the gene. So hypermethylation generally tends to turn a gene off. Hypermethylation or undermethylation actually activates the gene. So there's a mechanism beyond just the gene, which is epigenetic markers, which allow the turning on and off of genes, and that can cause severe diseases. Why nanopore? Oxford Nanopore data is information rich. Methylation information is something that we capture when we sequence a strand of DNA. the methylation signature is actually encoded in the signal that we capture. Because we have long reads in Oxford Nanopore, we are also able to haplotype resolve, the two different alleles coming from the two different chromosomes, and you can overlay the methylation information on top of phasing and be able to differentiate between paternally inherited methylation markers and maternally inherited methylation markers. And in this graph, you can see the blues at the top are essentially hypomethylated regions, as in loss of methylation, and the reds tend to show increase in methylation. And you can actually see between haplotype 1 and 2, there's a certain difference. There's a sea of blue on the top panel and a sea of red in the bottom panel showing differential methylation across the two copies of a particular gene. In this case, KCNQ1. We can phase, and I've mentioned phasing earlier, because there are long reads, you can then determine differential patterns of mutations across the long read and be able to phase between one haplotype and the other. And then it's not just over short ranges, because these reads are longer, you can actually phase across large loci. And then across this region that I show below, you can actually see long range phasing, starting from genes MPPL23 onwards to KCNQT1, and you can actually see phasing happening where you can show, a paternal haplotype being methylated, and further downstream you see a maternal haplotype is methylated. So this is all possible with Oxford Nanopore technology, and it comes with the sequencing. You don't have to do anything extra like bisulfite sequencing. Now, imprinting disorders. Methylation is critical and is for imprinting disorders. What imprinting means is that you take a copy of methylation state from one allele and transpose it on the other. And this happens when you get both alleles coming from the mother or both alleles coming from the father, or for some reason, both copies of a gene are hypermethylated. This example shows you two different syndromes, Prader-Willi and Angelman syndrome, which are both caused by differential methylation on the same gene, SNRPN. Now, this gene occurs in chromosome 15, 15q11.2 to be exact. And Prader-Willi and Angelman are actually very different phenotypes. So Prader-Willi is generally, Prader-Willi leads to eating disorders, hypertonia, obesity in later life. Angelman syndrome, on the other hand, leads to epilepsy, intellectual disability, ataxia, and so on. They're both caused by differential methylation on the same regulatory region. In Prader-Willi syndrome, the regulatory region is hypermethylated, and you can actually see a series of reds. And in Angelman syndrome, that regulatory region is actually undermethylated, so unmethylated, and you see a sea of blue. Now, just that selective turning off and off, on and off of genes seems to cause two different diseases, on the same loci, on the same regulatory region. So nanopore sequencing can detect these easily because, like I said, methylation comes free out of the box and is easily detectable. It's phasable. Now, these graphs also show different ways by which you can actually get Prader-Willi and Angelman. So you could have uniparental disomy, where both copies of the gene come from the same parent. You can have microdeletions and so on. So the net effect in these cases is that one allele or the regulatory region of one gene of this gene is methylated, and in the other it's unmethylated. More recently, there's been a paper which has been studying whether long-read sequencing can be part of standard clinical testing for Prader-Willi and Angelman syndrome. They started with 23 positive samples, used a long-read whole genome on PromethION 24 machines, and created a one-page report to visualize this. And their results showed 100% concordance with a known clinical test result. And this is what the one-page report actually looks like. So that especially look at, the region around the gene and be able to then identify regions of homozygosity or absence of heterozygosity, which is in the middle plot, which suggests that, both copies of the gene have been inherited from the same parent. In plot number C, you can actually see loss of methylation between all this all the CPG sites, which suggests that this is leading to Angelman syndrome. Loss of methylation in this case leads to Angelman. Increase in methylation results in Prader-Willi syndrome. So this is absolutely possible, and they recommend that long read sequencing be taken into the clinic and become kind of a standard for routine clinical care moving forward. What else can nanopore long read sequencing do? You can investigate repeat expansion disorders like Fragile X. These are difficult to identify with short read technology because they're triplet repeat expansion. So you have a pattern like GCC, 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 expanding or contracting, and you can have thousands of these repeat patterns. When you have short reads, you can't align them to the reference genome as well as you would like to. But long reads, it's possible because generally the repeat expansion is spanned by a single read or multiple reads that span the length of the expansion. So what you can do with long read technologies like nanopore is count and annotate triplet repeat expansions, identify the novel expanded allele, and identify, because you've got methylation information, you can identify if the non-expanded allele is imprinted with the same methylation patterns as the expanded allele. And I have a graph on this on the right-hand side, which shows, how imprinting happens. So you can actually see, on the right side, on the left side of the right-hand graph is the percentages of bases methylated. So in the top row, you've got hypermethylation happening, and in the bottom row, you've got hypermethylation happening across both haplotypes. So in some cases, you have imprinting happening where you have one copy of the gene that's expanded with triplet repeats and the other is not, but methylation plays a role here. So there is a method, another paper that I'm referring to by Sinha et al. This year in Nature Communications, they talk about an epimarker method. So they've created a database of 36 diseases and their episignatures, which can be used to quickly query a particular data set against, and that has characteristic methylation signatures for various diseases, including SMA. And you pass your data through epimarker, and it can come up with kind of a recommendation on or an identification of regions that might be being influenced by methylation changes and by epigenetic modifications. Finally, why nanopore? Right, so it's one assay, and it reduces the time between clinical assessment, sequencing, and interpretation of results. If you remember right at the beginning of my talk, I mentioned a diagnostic odyssey, which can take up to 5 years, 10 years even. You can cut all that short by a massive amount of time, and you can get results back in a week between clinical assessment, sequencing, and interpretation. So why would you want to make your patients go through that kind of diagnostic odyssey, even for clinical research specifically, right? Why would you want to take such a long time trying to diagnose a particular case which has gone into clinical research when you can reduce the time by a single experiment? What else does nanopore bring? So I've got a comparison here between different technologies and what they can and cannot identify. So if you've got short read whole exome sequencing or short read whole genome sequencing, you can at best capture accurately SNPs and indels. You can identify certain large copy number variants. You can identify mitochondrial variants. And, if you were to move to nanopore, you can identify them all, everything starting from SNPs to complex structural variants, genes and pseudogenes, repeat expansions, mitochondrial variations, plus methylation. So a single experiment, all the results that you're likely to need to identify a genetic cause for a disease is available with our sequencing platform. It's not just sequencing. We are working really hard on an end-to-end platform. So we have everything to go from library prep in a few hours to nanopore sequencing up to 72 hours to a data analysis platform. And for germline, we have a bespoke workflow called WF Human Variation. So you can go through an end-to-end pipeline just using nanopore recommended tools and nanopore technology. At the very end, you have the analysis pipeline, as I said, WF human variation, which will do secondary analysis, alignment against the reference genome, and call variants across various classes, including SNPs, structural variants, copy number variants, tandem repeat expansions, and methylation data. Once you've got that data set, you have to go through interpretation and annotation of those variants, which is where we've been working with partners, a lot of tertiary analysis partners that we've been working with, and Dr. Mohammed Udin is going to be talking about one of the partners that we are currently, starting to work with, Horizon Genome Arc. So variant interpretation basically involves annotating the results that you've got from the epitome WF human variation workflow with classic, database resources like population frequencies, annotations for genes, predicted mutations from ClinVar and dbSNP, and then be able to prioritize a set of variants identified in those VCF files based on your disease category, Mendelian inheritance, phenotype, and then be able to score them on ACMG category, and then finally be able to create a report for that particular case that you've been trying to interpret by basically identifying a set of likely causal variants or the causal variant that is responsible for the disease for which the patient or the case was sequenced. So we've got a complete package, therefore, starting with sequencing, library prep, sequencing, secondary analysis, and tertiary analysis with our partners, various partners that we've been working with. And you're going to hear from one of them immediately afterwards. And finally, just if I have to leave you with one slide and one message is Oxford Nanopore is not restricted to SNPs and indels like short read sequencing technologies. We can add additional features to your sequencing data, including methylation, structural variation, complex copy number variation. And I hopefully in this short presentation have shown you some examples and given you some insights on what you can do with our sequencing technology. And I shall leave you at that. Thank you very much for your time.
Hello, everyone, and welcome. I'm Mohammed, founder of Genome Arc, and I'm excited to present our work on building the AI-powered unified multimodal platform for scalable clinical genomics. So we are now entering the era of population scale genomics. Initiative like these, always wonder how we can analyze such a big data set in a reasonable time. So the UK Biobank, if you see, it's actually touching 500,000 samples now. The 470,000 exomes are available. Broad institutes are, almost exponentially sequencing samples. There are also Asia that are catching up with thousands of genomes. So almost all countries have their own population scale genomic projects. This scale demands platforms that are both powerful and flexible enough to manage, interpret, and integrate massive volume of genomic data. Although clinical adoption is slow, clinics and hospitals are integrating clinical genomics unit within their healthcare pathway. More newborn screening programs are announced over the years in different continents. And slowly it's getting a first-year test for rare disease. If you look at rare disease, genomics is extremely effective. Rare disease affect millions worldwide. Most rare diseases are genetic in nature. And we actually saw in OMIM, there are more than 5,000 rare diseases being recorded and documented. Children are disproportionately affected, and the typical diagnostic journey takes up to 5 years, which is also known as diagnostic odyssey. So we believe integrating this multimodal data will able to decipher those complex cases in rare disease that are currently not being diagnosed properly, or at least in timely manner, because the time is a big factor in therapeutic intervention as well. In oncology, the complexity is even greater. We are dealing with germline variants, somatic mutations, and liquid biopsy signals from ctDNA. The molecular insight of these tests are now analyzed together to guide both diagnostics and treatments. So we need a platform that supports this integrated view of oncology, because germline only constitute about 10 to 15% of the diagnostics yield overall in different cancer types. Somatic variants is very complicated to detect small clones, but you have to sequence with higher depth. And liquid biopsy still requires, there needs to be a tremendous effort in algorithmic development to identify what kind of cancer you can predict from ctDNA signals. And the interesting development of clinical genetics is that we are no longer working in genome data alone, because it's kind of limiting if you are only using genomic insight for diagnostic and therapeutics, because there are clinical symptoms data from patients, multiomics, single cell transcriptomics, and proteomics data. All of these molecular and clinical multimodal data needs to be integrated in a comprehensive and cohesive manner. And if we cannot integrate these multimodal data, we will not have a complete understanding of disease mechanisms, will also impact diagnostics and therapeutics. So one of the biggest challenge in this, large-scale clinical genomic era is not just sequencing, because sequencing is now pretty streamlined. It is actually the interpretation that is the biggest bottleneck for us. Even for some of the known cases, just to process the annotation, it takes hours and hours. So analyzing an entire genome with clinical relevance, when it is complex, because you're dealing with millions of variants, small and large, we need a system that go, beyond that timeline and return something very fast, so the diagnostic and intervention decision can happen in minutes, not in hours or days. So we here at Genomic built a solution. We call it Genomic Horizon, a multimodal platform for clinical diagnostics. It helps in many ways. So our platform offers ultra-fast analysis. Exome interpretation happens within 5 minutes, whole genome within 10 to 15 minutes. It works both locally or in the cloud, ensuring speed, scalability, and compliance with privacy protocols. It's encrypted. It's heavily customizable based on the need of the clinic or labs. And it's technology agnostic. It supports different sequencing technology. So Horizon actually integrates proprietary database, multiomic database. It connects with the patient health record if it needed to, and there is intuitive interface that is connected with our own AI agent that built within the integrating these data sets. So it is a unified multimodal platform built specifically for clinical genomics. At the heart of this platform is Horizon Intelligence. It is a intelligence that we built to explain all the different features. So Singularity is a feature that we built for rare disease diagnostics. It can analyze a single genome or a trio or a duo for carrier screening. Spectra module been developed for oncology solution, can do genome analysis for constitutive variant findings. Also, it can profile tumors, and also it can predict ctDNA signals. And the last module that we have is the SICRX, is the pharmacogenomics module that predicts the pharmacogenetic variants and also provides a range of drugs options if there is for different gene variants. And if you look at the clinical insight that Horizon provides, it's basically targeted towards rare disease and oncology. We use ACMG guided diagnostics protocol, and that is ultra-fast carrier screening for true premarital carrier screening or trio analysis, secondary findings. Tumor profiling is another important task that we do. Tumor somatic variant detection is an automated process when you upload the VCF. Liquid biopsy or ctDNA data can be uploaded to identify the ctDNA variants and its effect and what kind of cancer it might it predicts what kind of cancer might manifest. And there are a few other modules around oncology, which is implemented in Spectra. And then all these clinical features actually integrates the pharmacogenetic information. You can upload single cell transcriptome data from the tumor or proteomic signatures that will all be integrated into one intuitive platform. I want to share our experience on integrating Horizon with Oxford Nanopore technology using Oxford Nanopore's A100 GPU towers. What we observed that when we took the VCF as input from a long-read genome that constitutes about 5 million small variants, SNVR indels, and about 40 to 50,000 structural variants, it can annotate and predict the variant pathogenicity in 12 minutes, the whole entire genome. And it includes full analysis of the annotation, the variant classification, and also report generation. So all is in one cohesive way. So, and also when you are analyzing the whole genome, you are also in parallel can sequence the whole genome. You can do a LSK whole genome run in your P24 or P46 machine, and still can use Horizon to analyze the already sequenced VCF files. So this is an example where we have a rare disease patient using Singularity module within Horizon. We identified de novo variants. So because the de novo variants, if you upload trio data, probe and father and mother's VCF file, it can identify automatically all the de novo variants. So this is an example where we identified KMT2D variants that is a known causal variants for neurodevelopmental disorders. And this kind of rapid insight actually can accelerate diagnostics dramatically and also can help a clinician to act in a timely manner for therapeutic intervention. And you can see in the IGV shows that the variant is present in the proband, which is absent in father and mother. And we have another case involved a colorectal cancer patient. So traditional targeted genetic test failed to explain the cancer or why the toxic reaction was manifested after introducing chemotherapy into this patient. And our pharmacogenomic module from Horizon identified a DPYD deletion variant that is actually linked to the drug metabolism and known to have toxic reaction if the gene is mutant, or the patient is mutant for this gene. And we also found at the same time the cause for the cancer, which was not known. So we found a very interesting mutation in TCF4 gene. So these are examples where you can actually bring different kind of modules in one place and kind of interpret the data, because the clinical data helped us identify the variant that we are looking for in this particular patient. We'll show you how a clinical symptom can be used to generate gene panel, and that can be used in Horizon and link with the patient genome. So our tumor profiling. So the last case we just discussed was for germline variant, but Genome Arc Horizon also equipped with tumor profiling. So in our tumor profiling module, Spectra, we invested a significant amount of algorithmic development. Our platform can detect somatic variants, can classify pathogenicity. It profiles tumor mutation signature to assess the DNA damage. It constructs a full clonal tree for somatic variants, and like how many somatic variants in what clonal complexity. It can identify the tree-like structure for different clones and subclones. And it also can predict from ctDNA what is the cancer that might manifest, because ctDNA will be one of the biggest tests in future for cancer prevention. When you do super deep sequencing of a few targeted genes, whatever the variant you find, you might always struggle to identify the cancer type that might manifest with this particular variant. So in one of these use cases, we uploaded 53 kind of data from ctDNA, and we saw that because it's a pan-cancer gene, and we saw that particular variant have a 43% probability for lung adenocarcinoma, and the probability of other cancer types are also provided. So these are the features that actually comes just from the VCF files that you upload. And it's not just that. Genomics can be very interesting in terms of population-specific variants, because from the out of Africa hypothesis, each population actually have some bottlenecks, some rare variants they lost, or some rare variants become common. Even founder effect can be detected. So here's an example where we uploaded a custom database. And you can see on the diagram on your left that all the different ethnicities from Nomad there is no bar, there is no frequency of the variant, but the custom database at the end shows it is a common variant in Middle East, actually. So there are a lot of these type of common population-specific variants across the globe, and you can upload your population data into this platform, and it will integrate that population frequency during the pathogenicity calculation. So a lot of our modules have been validated from different sources, and we also looked at very complex cohort of rare disease and cancer cohorts. So this is one of the rare disease cohort where we saw that there is a yield of 36% using our software, using Horizon. And it identifies all the known variants and many novel variants. And we also kind of did a big test on the effectiveness and sensitivity of the structural variation pathogenicity prediction for rare disease and inherited cancer. And our ROC analysis shows overall accuracy of 96% in predicting structural variation pathogenicity. So here's a video that I want to show how we use the unstructured clinical phenotype data and predict how we predict disease and create a gene panel. So this is the agent we created using the large language model. So for example, you can use this panel, and you can paste or dump any unstructured data. It can come from your health record. It should have the symptoms of the patient. And once you paste the symptom data into the panel, you can update the page with this symptom. What it does, it will bring out all the HPO terms that is relevant to the disease. For this particular example, it is a Duchenne muscular dystrophy description we added, and it identified exactly that particular disease with highest degree of probability, and the the disease, and the disease gene it will identify that is associated or known. Not just that, it actually creates a gene panel. So all these genes that you see have different degree of probability. So I want to show you another AI agent that is built within the system. So it's within Horizon Intelligence. Apart from that phenogen module, the phenotype genotype module, you can actually search gene variant. So you can search gene names and ask for a function of a gene that is within your genome, and it will print all the description related to that gene in a very fast manner. So, for example, if you have a gene that you don't know what it does, because lots of viewers variants are impacting genes with function that we don't know. So this agent actually can identify all the relevant disease information. Not just that, so if you upload genome within our platform, the VCF files, it learns those variants, and it can find variants within these genomes by typing the variant location, just like that. So it should identify if there's any pathogenic or clinically relevant variants in this location of the genome for any of the patient. So, and what are those patient? What is the phenotype? What is the gene within this impacting this variant? It should identify all these in a small table. So having this option actually helps you analyzing. So this is a frame-shift insertion that has been tagged as pathogenic. In many other projects and individuals within this platform that you uploaded. So these are individuals that you have uploaded. So the language model learns those and identified. And if you want to know, so this also identified what gene it is. And if you want to know more about this gene, you can just follow up the question within the language model. So you can see how we are using clinical information and genome at the same time to bring the diagnostic yield. And this actually help us, shorten the diagnostic odyssey time. So in summary, what Genomic Horizon actually does, it automates the whole multimodal process. It uses tons of proprietary databases, and it integrates different modalities into one cohesive platform. The algorithms that we use are validated and secure. The large language models comes with many different agents, and we are constantly building different agents for different types of algorithm. And it's ultra fast. It's super fast. It's super secure. It's encrypted, and the way it integrates all these modalities, it will enable early intervention for rare disease and cancer prevention. So this is a disclaimer for Oxford Nanopore Technologies. These are for research use only. And thank you. So please let me know if you have any question or concern about Horizon.